is episode 267 of the Stem Cell Podcast, Embryo Morphogenesis, with Dr. Shankar Srinivas. Hey, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the podcast, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Shankar Srinivas, who's from Oxford University. He's on the podcast to talk about his research on the fundamental cellular forces that drive morphogenesis of mouse and human embryonic development. We've also got our usual roundup of recent highlights in stem cell news. That's coming right up.
But first, we'd like to remind our listeners about ISSCR 2024, happening July 10th to 13th in Hamburg, Germany. Join more than 400 scientists from around the globe at ISSCR 2024 to take part in a 4-day program. Learn more and check out the program at isscr2024.org. And indeed, we are very excited to attend this year, in part because, of course, we'll be recording at the meeting like we always do, and we always have a booth, and we're happy to meet up with everybody. But we're also putting on a live show this year with guests from folks who work with the International Space Station and do research in space, like yours truly. So it's something that's near and dear to my heart. We're very excited about it. So mark your calendars for Friday, July 12th from 6 to 7 p.m. over in Hamburg, Germany, and we'll see you there in person. All right, let's dive right into it. This is the roundup section of this show, episode 267. This is going to be a fun one because we've got a couple of back-to-back papers here coming from Jun Wu and colleagues. These are just a couple of high-profile papers that have been published in Cell recently. And if we're talking about Jun Wu and we're talking about his work, of course, we're going to be talking about blastocyst complementation. This is one of the things that he's really known for in his laboratory. In fact, he was here at Cedars-Sinai in my home institution just a few weeks ago talking about some of this work, actually not specifically what we're going to talk about here today, but in general, the area of interspecies blastocyst complementation, its potential, and some cool in vitro experiments as well where they're looking at competition between mouse and rat cells in the same population and how that can actually inform some of the work that they're doing in vivo. So let's get right into it. These are two back-to-back papers that I'll cover here. First is titled Generation of Rat Forebrain Tissues in Mice, coming from Jun Wu's lab. The second paper is a similar story using similar interspecies blastocyst complementation techniques, titled Functional Sensory Circuits, built from the neurons of two species. And Jun Wu is also a senior author on this paper. Also on this paper is his mentor, Juan Carlos Ipsuza Belmonte, over there in San Diego, but also Kristen Baldwin, who's one of the senior authors in this paper. Big picture, where we're talking about blastocyst complementation, being able to integrate the cells of multiple species during the developmental process. And in the first story, this generation of rat forebrain tissues in mice, I mean, this is a big deal. It just obviously it kind of shows in the title of the paper. They're able to, in a nutshell, generate mice that were null for their forebrain, basically complement the blastocysts of those mice with rat cells that were able to restore and generate a rat forebrain in a mouse. Pretty amazing stuff. But that's not all there was to it. The foundation of this story was this CRISPR-based blastocyst complementation technique, or CCBC, which they actually used to identify some of the critical genes, in particular HESX1, that are critical for the development of the forebrain. Okay, so they're able to show using the C-CRISPR rapid screening technique that this HESX1 deficiency supported the generation of the rat forebrain tissue in mice via interspecies blastocyst complementation. But upstream of that, which is of course the very high profile part of the story, upstream of that, they're able to validate that this C-CRISPR technique works intra-species as well. So, being able to complement mouse tissues too. But moving on to the xenogenic complementation part of it, The rat forebrain tissues in the adult mice were structurally and functionally intact. This is great. They're able to do some neuronal profiling here as well. They did some comparative analyses cross species, showing that the rat forebrain tissues actually developed at the same pace as the mouse host tissue, but importantly, maintained their intrinsic, rat-like transcriptomic profiles. It makes sense that the cells are from the rat, even though they're in the mouse host tissue, right? One thing to note here, and I remember, June was alluding to this, the speed of these cells in this single host, the speed of their development is impacted because, for example, the chimeric rate of the rat cells, in terms of the rate of which they're chimerized, actually ended up decreasing over the course of the mouse development. So it started off at a pretty high level of complementation, but over the course of the development of the mouse, the rate of the complementation actually diminished down to like 60% or something like that. And ultimately, this is just a benchmark story that's going to set up future studies, of course. This is not the first time, of course, interspecies complementation has been done. We've talked about Hiromitsu Nakauchi's work at Stanford almost a decade ago, where they were able to generate the rat mouse pancreatic chimera, and actually did that here in this story as well, to some extent, just as a validation of that their C-CRISPR technique is actually a good one in terms of identifying those candidate genes for complementation. But I think the power here is, of course, on the neuronal side of things. This, in the brain, right? Brain complementation interspecies, that really hasn't been done before to this extent. And of course, the C-CRISPR based technology is really, really exciting. So it's just, I think, setting up for future studies coming from the Junwu lab. And, of course, their lab is very collaborative, leading to the second story that we're talking about here. This is a collaboration with Kristen Baldwin's group, where they took this initial study a little bit further and validated it even further in a sensory context. Okay. So they did the same idea, this generating this interspecies neural complementation using this IBC, interspecies blastocyst complementation technique. And they were actually able to find that the mouse olfactory system can be accurately and reproducibly complemented by the rat olfactory system. Okay, so the rat neurons can actually restore information flow to odor processing circuits that are otherwise lost in this mouse host. So in essence, you're creating a mouse with the olfactory system, the nose, in other words, the olfactory neural circuit of a rat. Okay, so they can, I don't know, just like conceptually, that's so interesting to me. Imagine you're one animal, like say I'm a human, but I have the olfactory capability of a dog. That would be kind of exciting, right? To be able to smell things from very far away. I wonder what it's like to be a mouse with the olfactory capability of a rat. It's just a cool sci-fi concept to explore. They did this really cool Oreo cookie test where they buried the Oreo cookie in the mouse culture, the mouse living area, and the complemented mouse with the rat olfactory system was able to find that buried cookie. So it seems like it's working, even though this is a mouse. It has the functional olfactory system of a rat. And this is, again, just taking these stories a step further, that it's not just a developmental thing. It's not just that you're seeing cell complementation, you're seeing functionality too. You're seeing restoration of an actual olfactory circuit. And I think this has a lot of really interesting implications for future functional studies down the road.
Yeah, both these studies were, we got a lot to unpack here. And you took the summary down there relatively quickly, so we can chat about it. I'm glad, because I love blast complementation. That was my thesis, and I think it's as far as like experimentalism is concerned, it's a prime example, because it's one of those things where you're like, yeah, let's put these two things together and see what happens. And I think that approach, which is relatively descriptive and basic, has really evolved over the course of like 100 years since I was in grad school, to the point now that there's a lot more mechanism, a lot more nuance, the technical capabilities. The C-CRISPR, a great example, and the deficient embryos housing the trans species, all that has really developed in large part by Jun Woo and his collaborators. Um, so he deserves a ton of credit there. So this, to me, is not the ultimate culmination, but one of the many culminations of decades of research originating from this original basic idea of can you put these two species together and hope they develop. In my case, it was human and mouse. That was a big fail. But I was really gratified to see that the reason it failed, it succeeded to some degree, but it was very limited. And I was gratified to see that limitation is held. The cell intrinsic timing is of paramount importance. And our guest, Dr. Srinivas, I'm sure will attest that these things are baked in. And that's what we talked about in a lot of the studies we've covered and with a lot of our guests, the cell intrinsic timing limitation with maturation, etc. But here really presented in a very elegant way. Just to be clear here, though, in terms of the second study, in terms of the function, I just want to dive a little deeper because it's not like, and this is, I think, a key thing to address, from a philosophical standpoint and like an ethical standpoint. A lot of what's hampered, I think, progress in this research is some of the kind of bioethical quandary, namely what we got a lot of is, well, you're going to generate a chimeric mouse with human neurons. It's going to have human thoughts. And it's be an insult to human dignity. But, to be clear here, in this case, what this showed, it's not like mice can't smell Oreos, right? It's just that these mice were deficient fundamentally because their system was hijacked. Tell me if I'm wrong here. And then them being able to reinstitute that primal food-seeking behavior was more a testament to like they had functional neurons that were rat-derived. I don't know. Maybe I'm wrong there, but I think it's an important point to clarify because I think it can. I'm not saying it puts it to rest, but I think the idea of like a meta function being reconstituted by cells as the bit players, I think is a bit misguided. I think the idea of a dog having this amazing, capability is more about the organization of the olfactory neurons and the brain and all that stuff. It's not like, you replace all my neurons in their anatomy with dog neurons and I'm suddenly going to be sniffing stuff out. I don't know that that's not true, but I believe that it's not true. I think it's the organization and the anatomy and the meta that confers function. I think that's a really key point to make so that we can start to move forward with these brain studies. I mean, the lead in this story, and I'm not saying they were making this claim, but with all the xenocomplementation stories, there's a lot of like, we need organs, so maybe we can grow them in another species. And I think, obviously for the brain, that's never going to get off the ground. But also even for other organs, the pigs have kind of like made that a moot point, in my opinion. Still early days, but we got somebody walking around with a kidney from a pig now. It looks good, notwithstanding the 2 heart patients who succumbed ultimately to disease. But I think that the tech and the application there is very, very promising, perhaps more realizable than the xenogenic complementation stuff. But in terms of understanding the wiring, the function, the cell intrinsic versus, more collective attributes of cells from different species, also studying human cells in a tractable model that is, experimentally accessible, a laboratory animal, is going to really open the door. So there's so much in this story that is really a big deal. Probably the headliner, as you said, is that they finally got the brain. We're doing the xenogenic complementation, looking at the brain, not only looking at brain, but looking at functional cells in an animal, in a mouse, rat cells in a mouse brain. So really kicking open the door to a lot of applications, as you said. It'll be interesting to see how the C-CRISPR and all of the tech that's been developed here and over the course of all of these decades or multiple decades, how it's applied to unpack some of these kind of cross-species functionality questions. All these studies are ongoing and coming out at a rapid clip. As you said, Jun Woo, you tell me off the record earlier that Jun Woo came to Cedars. He didn't even talk about these studies that were in press. probably because, obviously embargo, but like still, he's got a lot more in the stable to trot out and share with us. So really looking forward to all that.
Yeah, absolutely. And I was definitely simplifying things a bit when it came to the behavioral complementation side of it. And how would you truly know? How would you truly know if those mice had the full olfactory capability of the rat? Like you can't exactly go out there and ask them. You can only do these third, one step removed behavioral studies. So it's certainly a lot of limitations here, a lot of things to improve on. I mean, for one, they didn't achieve full complementation. They didn't achieve full replacement of the mouse forebrain by the rat cells in either of these studies. The gradual decline in the overall chimerism, that's something else that they have to improve on. But I'm with you. I think this is a really cool study in part because it answers a lot of or alludes to a lot of evolutionary questions. We've been talking about neurodevelopment, the evolution of the neural system recently on the show. And you're right, June didn't talk about this specific study when he visited Cedars, but like I alluded to, he did talk about studies where they basically introduced pluripotent stem cells of multiple species in a single in vitro dish and saw that their competition rates were vastly different. And I think some of those purely artificial in vitro studies can help better inform some of these complementation studies and perhaps make them even better, right? I also totally agree with you. I think the big picture here of generating human organs in pigs and using interspecies blastocyst competition that way, I think that's I think we're still a long ways away from making that a reality. I think the current approach that's actually being put into patients, like you're alluding to, with the genetically compatible, immunocompatible pig organs and pig hearts and pig kidneys, that's happening. I think that's happening right now, and that's going to take a huge leap forward, and it's well ahead from the therapeutic applications of interspecies blastocyst complementation. But it's been a dream in the field for a very long time. And who knows, with studies like these that are kind of starting to break down those technological barriers to making that a reality, who knows what will happen within the next 5 to 10 years? We'll see.
Yeah, and I mean, you said, how do you know? How do you know in terms of conferring a species intrinsic function across species in the brain? I don't know how we're gonna figure that out or ask that question, but here we have the platform. So I do believe that someone is gonna ask that question, and I wonder, I'm so curious to see you put human neural cells in a mouse brain. Can you not augment to like make them smart, but will the behavior, will there be any behavioral influence that is not, degenerative, not a function of you breaking the system? Can you add function to mouse behavior, add a behavior? I mean, what a crazy idea. I honestly hope the answer is no. I'm looking for a negative result there because as soon as that comes back and it's positive, there's going to be a lot of people saying we should stop everything. So, really, really curious to see how, as you were alluding to, how are we going to ask that question? And I think we're on the doorstep now with having these, chimeric brains. It's going to be an amazing, amazing platform for study. Going back, I mean, here we're talking about the end game, the big questions about human brains and animals. Not that that's what they were actually doing there, but I want to take the other side of things, go back to the basics. The very first moves that cells make. Apropos to our guest today, who's really invested and interested in primary morphogenesis, primary, like the first things that happen. I got a story here I'm kicking off. I'll go back to back too. Not that I have a choice, but these stories fit together in the same way and are aligned with our guest today. So I'm really excited about the timing. The first one is about something that's really close to my professional interest here, vis-a-vis IVF, which is human embryo compaction. Okay, the first move that cells make besides just dividing. They human embryos, obviously we can't experiment with them as much as we would like, for good reasons. And for that reason, it's hard to really crack that question. How do these fundamental morphogenic events happen? Most of the time, we look towards these model animals, and you would expect that it's conserved, right? These are fundamental events, but we don't know that for sure. More recently, we have the embryo models, which we've said it before, have really kicked open the door to all this black box shining a light on the peri-implantation behavior of embryos. But still, we don't know how faithful those are to the real thing, right? So here we're talking about compaction, which happens on the fourth day of human embryogenesis, on the fourth day after fertilization, when the embryo is like between 8 and 16 cells. And you get this compaction event. Embryos, in the context of IVF, or in vivo, presumably as well, embryos that don't compact, or like compact not entirely and completely, they have been shown to have a lower implantation rate once transferred. So it's relevant to IVF. Compaction and defects in compaction has some clinical relevance there. And as I said there, as I allude to, the embryos can even compact like partially, incompletely. But why that happens, the molecular mechanisms that underlie that defect are completely unknown. But it's thought that it's primarily because of defective cell-cell adhesion. So that's the working hypothesis. And the group of Jean-Léon Mette, which sorry for the pronunciation, I'm doing my best, from the Curie Institute, and his group used micropipette aspiration in order to map the surface tension on compacting embryos. And they found that there is a fourfold increase in that tension at the interface of the cell and the medium, but the cell-cell interface, the cell-cell contacts, the tension stayed the same, pre-post compaction, same thing, versus cell-medium interface, fourfold increase, right? So based on that, they showed that cell-medium interface is critical and drives embryo compaction and the tension there, which is similar to what's been found in mouse embryos. But looking deeper in comparing mouse and human embryos, they found that there's different strategies that are used, with human embryos being less efficient. And that might explain, you look at mouse embryogenesis, pre-implantation, peri-implantation, it's like bulletproof. You get 10 fertilized eggs, you're going to get 10 blasts. Not so much in the context of IVF. So relevant there that the qualitative difference between human and mouse evolutionary explains things a little bit. They did inhibition of cell contractility or cell adhesion, cell adhesion, right? And showed that while you need both of these things in order to effectively compact, the contractility is really key. It is a key factor in controlling the surface tension. And when they looked at embryos that failed to compact, they found that the cells that didn't get in there, the cells that were excluded, had this defective contractility. So it's important. Okay, here we have the key mediator, I guess you would call it, a defect of which can really undermine contractility and compaction. Question, I don't know. Is that going to be something that we can hack? Is it going to be something we can rescue? Not sure. But I think fundamentally, the fact that we're able to verify some aspects of what we've learned in mouse embryogenesis pre-implantation is key. And also having a model here that's accessible, and a method here with this micropipette aspiration developed by the Metro Lab, I think is a really key advance. And I'm excited to talk with Shankar about this, because he has a vested interest in like the primary events. I mean, he had a very, I don't want to say similar paper, but he had a similar goal in a paper that we're going to talk about, I hope, with him, looking at early cellular events that mediate, in his case, also compaction. So apropos to our guest and a high impact paper from Nature. Arun, what's your take?
Yeah, this is good stuff. I mean, we're talking about evolution and the evolutionary differences in development between mouse and mouse, human, rodent, human, whatever. In our previous two stories from Jun Wu with the interspecies blastocyst competition, and this is building on that even further. I mean, like what we've talked about, and like other people have talked about here on the show, human development is so fickle, right, in terms of the efficiency of the process. In fact, just last week, Magdalena Zernica Goetz, who's down the road here in Caltech, actually was at Cedars-Sinai giving a great talk, and similar to the talk that she gave at the ICCR keynote last year. And she brought up that statistic where 60% of human embryos are lost before gastrulation even happens. It's such a fickle process, right? And so, in contrast to what we were talking about with interspecies blastocyst competition, which is, I think, far away from becoming a translational reality, I think some of the basic findings from this study can be used translationally directly, perhaps today, because the goal is to make IVF even better. And if perhaps you can harness some of these properties of embryo compaction and some of these observations, you might be able to do just that. So I think it's a really great story. It's a fundamental story, but I think one that has a lot of implications for IVF.
Absolutely. And I mean, as you said there, application to IVF, which is exactly what they're looking at, compaction, but also at a more basic level, just understanding these mechanical forces and how they govern behavior in a collective of cells is, I think, a really key approach that hasn't been, I think, emphasized. We look at the input and the output. We get one cell on the front end, we add some small molecules or cytokines, and then we get another cell on the back end. But I think the mechanics of how those cells behave in the midst of that is really key. And that's a good segue to my next story, which is also about cellular mechanics. And in this case, a little bit later in development, we're moving on to gastrulation, which is a key developmental event that has not really been accessible before these human embryo models. Now we're getting at it, but again, not really, not necessarily a perfect correlate of the real thing. And gastrulation, what it is how you get a single sheet of cells to form the three primary germ layers. And it's highly conserved evolutionarily. And it's how you go from, again, a single layered sheet, which is known as the epiblast, and to form the three germ layers. So just specifically in primates, the epithelium that ultimately becomes that epiblast, it persists for about a week post-implantation in embryos. And during this week, it expands in size. A lot of cell proliferation where it undergoes an expansion by about 10 to 20 fold in size. But the expansion in the cell number and the size of that sheet, that specific process, whether or not it contributes to lineage determination, is not known, because I don't know that many people have asked the question very specifically. In gastroid models, so again, looking at these embryo models, this is now approaching 10 years ago, which is nuts, but in gastroid models that are derived from human embryonic stem cells, in like a context of micropatterning, which was done first by my former mentor, my grad school mentor, Ali Hamati Bribanlu. And what they found is that these colonies would form ordered structures, but the mechanisms that drove that, while there's been an acknowledgement of cellular mechanics, the emphasis has primarily been placed on these diffusible factors, morphogens, right? BMP, active and nodal, all these TGF beta modulators are thought to be the major drivers of gastrulation. But in this story, and owing to a lot of other stories that suggest that cellular mechanics might also play a role, Jiwon Jang, who is at the Pohang University of Science and Technology in Korea, their group hypothesized that there's there's unique morphological features in the epithelium that drive lineage specification, right? Specifically, in this single layered epithelium, you have proliferation, right? But once you reach a limit, you get local cell crowding and you get extrusion, all right? Just looking at it, that's what you see. And there's a lot of evidence suggesting that cell crowding is a critical role in driving differentiation of cells, not just in making them to outbud. And this has been shown in many different contexts, including in the development of the zebrafish heart. There you go, Arun. It happens in the heart too, and it has been attributed to multiple factors. But most of the studies look at YAP, TAZ signaling, as well as piezo-1, and piezos, as we know, mechanical modulators, as well as YAP-TAZ. But in this story, Zhang Lab set out to look for additional factors, and they found one. They found that this pretty, well, a gene that only has been described, or mostly been described in the context of oncogenesis, ETB4, it actually links this mechanical process with gene expression. They showed that the cell crowding led to a modulation of ETB4 expression. And they went really pretty deep here. It's a Nature Cell Biology story where I think they really did a good job of unraveling the molecular and cellular mechanisms of this process. They showed that it's ETB inactivation that happens. So you get cell crowding, and then ETB4 is inactivated, and so you de-repress this neuroectodermal differentiation potential, right? So, and again, mechanistically, they show that this is based on this integrin actomyosin pathway that gets inactivated once the cells are crowded. That blocks endocytosis of these FGF receptors, and that is what leads to the decrease in ETB4, protein stability by ERK inactivation. I mean, they went so deep with the signaling and transcription factors, and then they even add a little mathematical modeling on the top of it all to affirm their experimental results, showing that cell density modulates the spatiotemporal ETB expression pattern and ultimately converges on lineage specification and development. So a lot of moving parts in this story, but I thought a really elegant demonstration at the mechanistic level of how you go from like a physical process and via these molecular signaling cascades, and that leads to not just a shape deformation, but that also is coupled with lineage differentiation. So while before it may have been thought that the spatial reorganization changed the morphogen gradients, here they're showing it's a direct contribution that happens hand in glove, and they show some of the mediators of that. So a really nice, elegant, and deep story from the Zhang Lab.
Yeah, beautiful, basic mechanistic story here that has, I think, a lot of implications. We see it all the time whenever we're doing differentiation, cell culture, pluripotent stem cells. Cell crowding and cell contact is so important, so critically important in that context. I mean, even me every single day doing cardiac differentiation, that cell confluency is one of our factors that we have to check off during our differentiation process. Ideally, we want to have like 80 to 90% confluent cells to perhaps have the best shot at getting a successful cardiac differentiation. And we know the fundamental principles driving that to some extent. We've fleshed that out through, like you mentioned, cadherin contacts. There's a billion different ways that cells sense mechanotransduction and then cell crowding in that way. But I think the cool part of this story is you have a new candidate. You have a new member in that family of factors that can influence mechanical transduction in this ETB4. And it's a candidate that we haven't really thought about a lot. I've heard of this before, this transcription factor that's critically important in oncogenesis. And I think we've actually even covered it on some other stories before. It also tells you the diverse role of a lot of these factors and a lot of these transcription factors in influencing cell dynamics. You actually mentioned YAP-TAZ. That's honestly one of my favorite signaling pathways, and I know it's not important in the context of this story, but it is one of those factors that's also important in signal transduction and cell crowding and influencing cell differentiation. In that way, YAP-TAZ has this unbelievable phenotype when you modulate it, where you, especially in the context of the heart, and I'm sure you remember these stories from a few years ago, where you knock out one of the YAP-TAZ members, I forget, and you have this massive cardiac hypertrophy phenotype, like these 2x bigger hearts than normal. And so it just tells you the importance of a lot of these transcriptional elements, transcription factors in regulating cell dynamics, not just in vitro, but also critically in vivo. And it's cool how we're, even in 2024, trying to figure out what are these critical factors in regulating these processes. So great nuts and bolts, basic story in nature cell biology here.
Yeah, I'm really glad you mentioned that too, the focus on YAP-TAZ piezo. I don't think I emphasize that enough, because you hear any kind of mechanics thing and the gravity of those pathways, it draws you to it. But here I thought they took a really, unbiased approach, which was great, using, again, just a cell culture model as their lead. They did this analysis of crowded embryonic stem cells versus not crowded, as you were alluding to there. And that's how they set upon this target of ETV4. So again, as you said, there's all these studies that have really, dug deep on those known pathways. It took a lot of courage, and I think a smart approach here to arrive at this novel candidate. And we'll have to see how that plays in other systems, because morphogenesis is everywhere, not just primary gastrulation, but throughout development in all tissues, and even in the context of organ regeneration and recovery from injury. So not wouldn't be surprised if we saw ETV4 implicated in some of those other processes, and I'm sure this group and others are looking in that direction. Amazing stuff, the mechanics. I think as in vitro scientists, I would say, I'll go so far as to say we work primarily in vitro. We get so focused on these more simple pathways, but the meta is so much more fascinating and interesting. And these embryo models, the studies I talked about in terms of cellular biomechanics, your studies about interspecies complementation are all really about that idea. And a real expert in that field is Shankar Srinivas, who we're going to talk to in a minute. Before we get there, though, I got a quick message from STEMCELL Technologies, who'd like to introduce their one-stop resource for researchers who are looking to use organoids in their experiments. STEMCELL's Organoid Information Hub provides scientists with instructional videos, educational webinars, expert interviews, technical tips, and curated publications to help researchers set up and optimize organoids as a research model in their own labs. Learn about organoid culture from the experts at STEMCELL. Visit www.stemcell.com/discover-organoids. All right, you guys, with us from Oxford University today, we have Dr. Shankar Srinivas, who is Professor of Developmental Biology in the Department of Physiology, Anatomy, and Genetics. That's based in the Institute for Developmental and Regenerative Medicine at Oxford University. Dr. Srinivas is also Zeitlyn Fellow and Tutor in Medicine at the Jesus College in his teaching role. In the Srinivas lab, they use mouse and human embryos as model systems, and Shankar's group looks at the control of patterning and morphogenesis during the establishment of the anterior-posterior axis, gastrulation, and early cardiogenesis. Dr. Srinivas, thank you so much for joining us on the show today.
Thank you very much, Dale, for having me here.
It is our pleasure. Let's start off by asking you about your general research. That little capsule I gave, I don't think does it justice. You're focused on arguably the oldest and most fundamental question in developmental biology, which is how does the complex patterning of an organism emerge from a single cell? But you leverage some of the most advanced tools and technologies toward that end. Elaborate a bit on what the overarching themes of your laboratory are.
Well, as you said, I guess it's the fundamental question of what shapes our body. How through this almost magical process we go from an egg which doesn't seem to have any features, and certainly not the features of the fetus that we're used to, a head end and a tail end, or left and right side, or front and the back. How do those things emerge almost as if by magic? But obviously, we as scientists know that it isn't magic, that it's scientifically tractable. And the example I always give is of a chicken egg. It doesn't look anything like a chick, but you put it into the incubator at the right temperature, and it will always give you a chicken egg. And importantly, though we take this for granted, you only get a chick at the end of the incubation. You never get a puppy or a kitten. So again, this is baked into the chemistry of the egg, of the embryo. And so it is, almost as a mantra, I had to say this to myself. It is scientifically tractable, though I'm not sure in my lifetime we'll exactly know how, but that's the basic goal. How does the overall shape of the body emerge? How do the genes, how does the material, genes, proteins that build the body, how do they work together to give you this shape reproducibly?
Yeah, if you think about it that way, the fact that a chicken egg only gives rise to a chicken and not a dog, it's almost like development is miraculous in some ways, right? All these processes work together in unison to ultimately give rise to a functioning organism most of the time. And it's no secret that we're really in the midst of this renaissance. I don't know if you want to call it a renaissance, but really almost a recent revolution in developmental biology here in the stem cell field in particular, in large part because of these new model systems that have been developed to study early human development in particular. And we've chatted with many of these folks and leading these labs around the world. And, as somebody who's focused on the fundamental mechanisms actually driving early embryo development, could you actually talk about the general utility of some of these new model systems? We're talking about these like blastoids, gastruloids, pioneered by various folks around the world, and maybe how they can be best used to approximate and study these early developmental processes. So, how useful are these systems?
Yeah, absolutely, Aaron. I mean, of course, they're fascinating because, like you said, they recapitulate the miracle of developmental biology. And of course, they're more tractable. One aspect, obviously, is the fact that you can get the different cell types. But I guess the area that I'm most interested in, the morphogenesis, and that's an area where some of these in vitro models are perhaps less good. They're very, very good at generating the differentiation of the cell types, but of ordering them. But nevertheless, they show a remarkably spooky resemblance to the actual embryo. And as you described, the technology is getting better and better. So it's probably just a matter of time before they do give you really very good reproducible embryos, or things that appear to look like embryos, and maybe they function like embryos in vitro. And so already, I think they're interesting models, and they're very useful models. But for morphogenesis, I think there's still a little way to go. And I have actually slightly other more broader views about development that kind of relate to this. So, and one of the things is that it always has struck me that embryonic development in different organisms, they always find a way to get the job done. So, you have, even at the very pre-implantation stages, embryos from different organisms that look pretty different from each other, like the inner cell mass of a mouse or a human embryo, it's asymmetrically positioned, but that's not always the case. Sometimes the pluripotent inner cell mass will be uniformly spread beneath the trophectoderm, etc. But ultimately, it still works. The way the amnion forms in different organisms is completely different, but at these early stages. But ultimately, it still forms a functional structure. And so for me, that poses an enormous challenge in the sense that, as a counterpoise to what I said in the beginning about you always get a chicken, but never a puppy or a dog, which seems to suggest that it happens in a controlled manner. Within a single organism, it happens within a controlled manner. But then when you can compare across organisms, there seems to be a tremendous amount of flexibility, and it's hard for me to square that circle. The flexibility across organisms, whereas in the almost dare I say, determinism and within a single taxon. So yeah, so for me, that's a bit of a challenge. So in the context of the in vitro situation, there's then that added issue that you are playing with something that is ultimately very labile, which you can mold, and to give you an outcome. And of course, we're selecting for an outcome, something that looks like an embryo. But is it possible that you may get to that outcome through through a variety of different routes? And so the route that's taken in vitro might be different from the route that happens in vivo. And so that's a point that one has to always keep in mind.
Yeah, I mean, that's so many ideas there. And I think that's what the embryo models have done for the field, is they've kind of kicked open the door to all these borderline philosophical questions. I mean, you just described there how you can have, many ways to skin a cat in terms of like primary fundamental elements within a specific species that, totally unrelated to a different species, but a similar endpoint when you look at like the fetal stage of that embryo. And in terms of the limits of our understanding, for me, not that I get it, but I understand why it's so hard to get it, I guess, because you have to overlay like, eons, numbers that we can't even conceive of years of evolution, right? That happened each in these little silos, pretty much, for all these different species. And it's hard for us to, I guess, put that into context, all the time it took to skin that cat, because we're looking at them all side by side, right? So there's so many elements like that where we can't even really understand the path that these I don't want to call them organisms, but these species or evolutionary, branches took to get to that endpoint. And as you're describing, also the kind of gap there between the in vitro and the in vivo and the fidelity there, it makes me wonder. Getting back to a paper that you published almost 3 years ago now. We just talked about last episode about this Chinese group from a Chinese paper that applied spatial transcriptomics about an early stage human embryo, similar stage as to the stage where you were the first to do single cell transcriptomics on a very early stage human gastrula embryo. And, in those intervening years between the single cell and the spatial, adding literally another dimension to the analysis, there's been all this progress that we were just talking about in embryo models generally, but specifically in human embryo models. But as you said, the gap in terms of the lack of the implantation process and how we're selecting for certain outcomes in vitro versus where there's all these compensatory mechanisms that go on in vivo, where like an embryo can undergo all these insults and then it'll compensate and fill in and look, like there was never an issue at all. So there's a huge gap there in terms of the technical capability. And I wonder, I'm putting it to you, particularly because they develop in the absence of uterine implantation. Do you think we're ever going to reach a point where we have like a kind of singularity, as I alluded to in the last episode, where we have cultured human embryos that are aligned, at the single cell level in terms of identity and anatomy with a bona fide embryo? And I really think that's a high bar to set. But if we were to get there, what would it take? Or do you think it's not going to be possible?
I think it's possible. I think it's probably certainly by the end of the century. Maybe that's a bold statement. There's still 75 years. That's a long time. At the rate we're going, I think it's entirely possible. Certainly, when you said at the single cell level, at the transcriptomic level, that's, we're already close at various stages, but recapitulating the entirety of development, no. But but that's what I mean. I would separate out the cell differentiation, cell identity, generation of different cell types from the patterning of the morphogenesis. And it's always going to be easier to generate the different cell types. And it might be harder in vitro, certainly, to generate the morphology. But both of them, I think we will achieve by the end of the century. It's a matter of time. We're making good progress. The question is whether we'll do it as reliably. I mean, of course, even in humans, most fertilization events don't result in a viable embryo or fetus. But I think even in humans, it's still a much higher percentage that come out as a viable baby than in vitro. And so there'll be that other aspect of making the reproducibility and the reliability of the process, because in vitro, there's still so much variability. And that's really very weird. I mean, that must tell you something about the fundamental biology, because of course, the culture conditions, it's uniform when you have multiple gastruloids or blastoids, et cetera, in a dish. They're all experiencing the same chemical environment, but there's still variability. So, and of course, if you start with genetically identical starting material, so where does that variability come from? Whereas in utero, it appears to occur much more reliably. In a mouse, certainly the majority of in a laboratory situation, the majority of fertilized embryos will implant and give you a newborn baby. But yeah, all of that being said, I still answer to your question. I think within the end of the century, and maybe argue some people probably in the field will say, "Are you kidding?" Hopefully by the end of the decade. I think the end of the decade is a push, but certainly by the end of the century.
Yeah, I think it depends on who you ask. I mean, I'm sure there's certain very optimistic folks in this field. I think some of them we've actually talked to here on the show who will argue that maybe it is the end of the decade. So we'll wait and see, I guess, right? I mean,
But Arun, I think Dylan set a high bar, said something comparable to say a human or a mouse fetus.
Yeah.
In morphology as well. And I think the morphology is, definitely not by the end of the decade.
Right. Yeah. Stay tuned. I mean, we're making a lot of progress. And if you just think about where we've been over the last decade from 2014 till now, it's incredible. It's just almost exponential in some ways. I mean, you also talked about in your response there, you talked about the importance of studying different cell types and generating different cell types, and how we've been able to approximate some of these developmental processes and recapitulate them in a dish for a couple decades, many decades now, to make things like, say, cardiomyocytes in mass in vitro, again by harnessing the same developmental principles, pathways, signaling pathways, all that. And now, like, my undergraduates who work in my laboratory can make iPS cardiomyocytes by the billions because these protocols have been so refined. But there's still a need to still dig deeper into some of those fundamental mechanisms of those protocols, not just to make them better, but just for the sake of understanding, right? And along those lines, you actually had a paper a few years ago, the science paper, which as a cardiac biologist, I thought it was groundbreaking. A lot of folks in the cardiac development field thought it was truly revolutionary. I mean, your lab was able to characterize a common progenitor pool that gives rise to cells of the epicardium, which is the outermost layer of the heart, and also the myocardium, which is the muscular layer that does all the hard work, right? I mean, so could you tell us more about this particular study, maybe the underlying inspiration behind the work? And we talk about the utility, right, of applying some of these discoveries towards in understanding differentiation in vitro and all that kind of stuff. Could you also talk about the ultimate utility for this work and maybe studying cardiac development and disease?
Well, thank you, Aaron, for those kind words about the paper. We found it great fun, and it was really, it was really great. So it really started in the heyday. I would still say perhaps in the heyday of the start of the single cell revolution, so to speak, and caught perhaps the tail end of the initial peak revolution. So we use SmartSeq too. So we started doing the work before 10x was widely available, but it was better because we got better depth. We combined both the thing I like about the paper a lot, from my personal perspective, is it isn't an atlas of the various cell types, which there are lots coming out at the time. Even now, there are lots of atlases coming out, and they're very, very useful. But we were trying to address the biology of it, and so we combined the transcriptomics with a lot of detailed imaging to try to map anatomically where the different clusters were. So Richard Kaiser, the first author, the co-first author on that paper, when he was doing these HCRs to try to identify the anatomical position of the various clusters, there was this one cluster which was a bit odd, and it was in between in its transcriptional state. It was difficult to annotate exactly what it was, and he was very, very good at pinpointing where it was, and he noticed that it's in this region which overlies. If you just do a normal, without doing a confocal, if you just look at an object from the outside, it might look like it's the cardiac crescent, but it's actually a little bit of the mesoderm, which is that's what we call the juxtacardiac. It's at the confluence of the extraembryonic mesoderm of the yolk sac and of the somatic and the splanchnic lateral plate mesoderms. So that was the start of the story. I still remember, actually, I was on sabbatical in India at the time, and we still had these regular meetings. And Richard said, I found this place, this cell type. I'm convinced it's a different group of cells, etc. And so then we followed through and made a reporter mouse, a Cree driver mouse, so that we could do a genetic lineage analysis and show that it contributed both to the myocardium as well as to the proepicardium, which contributes to the epicardium. So I think, it was a very, to us, a pleasing entry. In fact, that was the first single cell paper we had. And it was very pleasing to be able to bring these different newer technologies with slightly newer, older technology like HCR. Even now, HCR is actually much more common than it was even just 5 years ago. Though, of course, HCR has been around for quite a long time, but it really enabled us, because to try to anatomically position things, you needed the intersectionality of multiple gene markers. HCR really helped us because you could do multiple orthogonal fluorescent probes for multiple genes, and then, of course, image them with a confocal microscope so you could get the three-dimensional location. In terms of what types of things it can help us do, like you alluded to in the beginning, we can culture these different cell types, but some of it is we still don't understand at the molecular level exactly necessarily what is going on when you change the different components in the media, etc. And hopefully this will help us understand the standard stuff, give us a better handle on the molecular genetics, the trajectory of differentiation. More importantly, though, I guess it helps to understand how the first heart field cells that contribute to the myocardium, they're in this pool. We still don't know whether they're bipotent or if it's a population of unipotent cells, the juxtacardiac field. But where their feet sit, it helps us understand where they came from, how they may be intermingling with cells of different cell types. And a little bit also about the morphogenesis, which still, of course, is dear to my heart, where these cells, and sorry, there's another thing which I really love and talking about, and it's fascinating. The heart starts out ahead of the brain during development, literally. Temporally, it starts to develop before the brain, and spatially, literally, because of the way the tissues are arranged, it is literally rostral to the neural folds. And then you have this thing called rostral folding that brings it to its more inferior position within the body. And of course, at this stage, again, sorry, it's a bit of an aside, the embryo is a flat disc, and it's this type of rostral folding, caudal folding, and lateral folding that brings it together to form a tube. They all meet, of course, at the umbilicus, your belly button, and then your mouth and your anus form by cell death, and that's what gives you the tube of your GI tract. So to get back to the heart, it's this type of it helped us also understand how the rostral folding can contribute in bringing this juxtacardiac field more inferiorly and positioning it with respect to the heart.
So first, I'd like to say I like the way you provided the context there at the beginning of that answer, that the peak-seq, or at least single cell, was about, atlases and really unveiling all the diversity at the single cell level. And then, I think now the next wave was applying it. And of course, these things are all contemporaneous, and still atlases coming out, and there was mechanism coming early. But conceptually, I think there's that neat idea that there's the atlases, then there's the insight, and at the end of all of it is mechanistic understanding of the system. And then later on that answer, you started riffing about the mechanics, cellular mechanics, and I saw you light up. I mean, that's your thing. That's your wheelhouse, and I can tell that's your passion. And it gets to that fundamental question that you started the interview with. You know, how do you make shapes? And your work, a lot of your work is really rooted in that idea, the biomechanics of cell behavior, how it governs patterning, but at a really root level. You know, for example, the mechanistic underpinnings of blastomere compaction. You know, that was a big story. Yours, primitive streak formation, big story. Yours, just general morphogenetic processes and how the cells make those moves. And I get it. As someone who got hooked on science by watching radial interculation of xenopus embryos, when you start talking about rostral and all that stuff, I get excited too. I totally get it. But for some of our listeners who might be interested in how that, you know, cellular biomechanics actually translates to kind of, I don't want to say therapeutic endpoints, but it's critical, you know, understanding cellular differentiation. You got to understand really at the root level how these cells behave. Can you kind of spell that out for us and our listeners, how cellular biomechanics and shape, anatomy, all that stuff can inform how we understand and therapeutically apply pluripotent stem cells and their derivatives?
Sure. I mean, it gets back to the point you raised earlier about the in vitro embryos that are being made, embryo models that are being made. And, you know, I think arguably, I think it's fair to say it's probably because our understanding of the molecular genetics of differentiation is more advanced than our understanding of the morphogenesis, that we are able to get a variety of cell types. We know a little bit enough to be able to put the right components in the media and et cetera to get the variety of cell types. Though, of course, much of it is also still through self-organization. But we still don't understand enough about the molecular basis for the morphogenesis to be able to control it very well. So if we are, you know, if we want to make, say, an entire heart in vitro, we need to be able to control the morphogenesis to the high level of precision to be able to get that type of shape. And ultimately, the function structure is so closely linked to function, and of most organs, not just things like the heart. Kidneys, lungs, you know, the branching architecture is integral to their function. So the morphology is absolutely central to the function of these structures and the organs. So that, I would say, is the main utility or driver of the need for a better understanding of this area of the morphogenesis. In terms of what actually controls and contributes to morphogenesis, I mean, it's a complex and huge field. I mean, there's some of the things you spoke about are things like the cell migration. But there's also the forces that are generated by cells, which lead to the buckling of tissues. And that has to be coordinated with the movement of cells so that you get, you know, a properly formed organ or embryo. But another thing which we're looking at more and more now is not just about the generation of forces, but also how how tissues respond to forces, in the sense that you can get shapes by applying forces asymmetrically, like say by poking a ball of dough or something of that sort. But similarly, but equally, you could have symmetrical forces that if your tissue if the biomechanical properties of your tissue is heterogeneous, it will respond in different ways. So if a region of your tissue is very stiff, the same force applied on a table versus a sheet of paper, it's going to cause a different degree of deformation. So by patterning the stiffness of tissues, you can get morphogenesis, and that might give you a better handle or better an easier mechanism for giving you long-range global morphogenesis. Because we already know of examples where you have gene expression patterns, like, you know, for example, the segmented pattern of expression of genes in Drosophila. There are ways of getting segmented patterns. So in addition to them, say, driving patterned cell differentiation, so cell type specification, what if they also, and you know, not what if, they almost certainly will also feed in to the biomechanical properties of those tissues, and they could then be leveraged to give you different responses in different regions of a tissue to the same global forces. So those are the type of ideas we're trying to pursue in the context of morphogenesis and how the control of morphogenesis. And if I may, of course, other aspects, since you mentioned also the single cell work, is there's a huge void. I mean, our single cell approaches, they're really very helpful in defining cell states and trying to make a sense of the trajectories cells follow when going from one cell type to another. They don't tell us, it's still hard to make many conclusions from them about morphogenesis. And that's an area which would be really a great, if we could find ways of linking the transcriptomic state of a cell to its morphogenic behavior. And that tends to be, of course, indirect because you had to go through the proteome. Much of the behavior is at the level of protein modifications, etc.
Yeah, I mean, I think the idea of understanding the fundamental biomechanics of cell behavior and morphogenesis has a tremendous applications, not just translationally, but, of course, for understanding the basic science. But one thing we've actually talked about a lot here on the show is bioengineering, and how bioengineering is actually starting to be intersected with developmental biology and some really unique applications. For example, there's this organ-on-chip technology, and we actually have chatted with folks like Matthias Lutolf, who actually had an amazing paper recently where he was looking at oncogenesis and modeling those fundamental processes of oncogenesis, which of course has parallels to development as well, right? I mean, what is oncogenesis if not development gone wrong, right? But they were able to study some of those fundamental oncogenic processes in this bioengineered organ-on-chip system, which is, really, really cool. Other folks have been able to use these chip technologies and other technologies like them to study morphogen gradients in vitro and how their influence on developmental timing and other related processes as well, stuff that we're talking about here. I also read your group actually has also developed some 3D models of developing embryos. So maybe you could talk about that area of how we can intersect bioengineering with developmental biology to model early development in a superior way.
Yeah, I mean, my lab personally is at very early stages. So we haven't really made it enough of a foray into it yet. But what my views on it are very aligned to yours, and I guess in the sense that it's a remarkable area, and I think it's an area of tremendous growth potential, not just for therapeutic reasons, but you could imagine, already sensors for chemicals, etc. In essence, an intersection where you leverage the strength and flexibility of known biological cell types and interface them with electronics. And I think that is the big challenge, would probably be a big challenge that there's already developments there, how to get, in essence, an interface to the electronic world that we have already existing, so that you can, say, feed the data through, say, a Bluetooth transponder and have it beamed to a receiver of some sort and act on that type of data. But I think there is, like you say, a tremendous amount of developments there in developing these in vitro things. In the context of developmental biology, of course, I guess there are additional models that we can then use to try to probe the fundamental principles of developmental biology. But again, that gets us back to the thing I was saying at the beginning, my personal concern in a sense. I don't know if it's a concern, or maybe it's, one should celebrate the diversity of different strategies. But I get the sense that different cells will find a way, but it may not necessarily be the way that it happens in a particular organism. And so if you're going to use that to, say, develop drugs for drug screening, it may not be suitable, or it may not give you an accurate response. It may not be an accurate tool. So I guess it depends on the questions and the questions that you're using it for, as in all things.
Yeah, and I mean, I want to emphasize that point because I'm the biggest offender when it comes to asking kind of, I'll say it, just dumb questions like, hey, are we ever going to be able to make an actual mouse with in vitro? It's like, yeah. And I asked it to you and you say, yeah, it may take a century. And yeah, you answer the question. Thank you for that. but I'll admit it's aggravating. It's annoying to ask because at the end of the day, I mean, the elephant in the room here, and you just alluded to it. Maybe you'll get to the same endpoint, but you got to go a different way because the elephant in the room here is the vasculature, right? I mean, how the heck are we going to recapitulate a full vascular system to get any tissue at scale? And yeah, that's probably why in vivo development is so robust that you could hit it with a hammer and it's still compensate and everything comes out okay because it's a living system that we can't really approximate. I have no idea how we're ever going to approximate that. So I want to apologize for asking the dumb questions because, as you said, it really depends on the application and the utility of these models. We're not trying to make humans, obviously. We're not trying to make whole animals. We're trying to model these fundamental processes that we have not been able to observe before. It's been a black box, and now we're able to shine a light in there. And I think you appreciate that more than anything. But I will say this. You know, if we are going to find a hack to get around that elephant in the room, the vasculature, it's going to happen at a place like Oxford, which is synonymous with elite education and scholarship. Just some fun facts here. Found like the oldest school ever, right? And founded almost 1,000 years ago. Like 30 prime ministers come out of there, heads of state from all over the world, kings, sultans, presidents too, Supreme Court justices, all the medalists you can ask for, Nobel, Fields. The biggest names, Einstein, Hawking, Hubble, Huxley, Schrodinger, Tolkien even, you know, Hobbes, philosophers, Locke. List goes on and on. But Oxford also has a pretty long tradition in the study of developmental biology and stem cells. Maybe not 1,000 years, but like at least 100. Tell us a bit about what it's like working at an institute with such a deep and expansive pedigree and some, you know, aspects of that experience that, you know, you wouldn't find elsewhere.
Well, no, it is fun and it is a privilege. I mean, so for example, we have colleagues like Liz Robertson here, who's one of the early leaders in developing embryonic stem cell technologies and making the first knockout mice, etc. When she was in Columbia and Harvard. So it's a vibrant environment. I also remember when you spoke, Dylan, about the long tradition. When I first came here to set up my own lab, Richard Gardner had his lab. He was still working here. Chris Graham had his lab and still working here. And for me personally, it was actually a pretty poignant moment when I gave a seminar. They were both there because in terms of personal relationships, Chris Graham, my PhD supervisor Frank Costantini, did his postdoc. He and Liz Lacey, they made the first transgenic mouse to go germline when here in Oxford in Chris Graham's group. And my postdoc supervisor, the late Rosa Beddington, she worked with Richard Gardner in Oxford, along with Ginny Papiano. So it was almost like both my, the grandfathers of my PhD and my postdoc academic grandfathers are there. That's it's been very good. In terms of more generally, Oxford is a nice place. It's it has a long history. It has all these quirky traditions. So in that sense, it can be strange, in the sense that the superposition of very modern technology and as well as long-standing traditions. So right now, actually, I'm in college. And my college is one of the middle-aged colleges. It's it's from the 1500s, so it's relatively young. But we're surrounded by interesting architecture. But my lab is in a brand new building, the Institute for Developmental and Regenerative Medicine, so it's got state-of-the-art facilities and things of that sort. So I guess you get used to it. And you start not taking it. Perhaps I don't think I'd take it for granted, but you get used to it. And but it's and from a practical perspective, actually, it's a good size in the sense that it has a life. There's things you can do outside the university, and though it is a university town, it is a bit bigger than the university, and there's more to it than the university. And in fact, there are two universities: the University of Oxford and Oxford Brookes University. It's also manageable, so that you can get from one end of town to the other on a bike in 15-20 minutes, and so you can get anywhere. and there's lots of green spaces, so it's lovely. And you have pastures in the middle of town. You mentioned Tolkien. C.S. Lewis is also, Oxford is also known for C.S. Lewis, wrote the Narnia books. And so, I sometimes walk through this floodplain, which has these, the lampposts, which, was, in the very first book, the witch, the witch, the lion, and the
Lion, the Witch, and the Wardrobe, yeah.
Wardrobe.
the Lion, the Witch, and the Wardrobe. Sorry, thank you. There's this lamppost, etc. It's I'm told it's modeled on those lampposts going through this floodplain.
Wow.
That's so much fun. I mean, there's so much history there, so much iconic influence of that place on literature, science, education, you name it. I mean, that it's a time scale that we can't really comprehend here in the US when you're talking about buildings that are, multiple centuries old, half a millennium old. But really an incredible place to be. And, one of a number of incredible places that you've been to during the course of your scientific training. You, of course, started off in India before heading to New York to work with Frank Constantini, like you alluded to, at Columbia for your PhD on kidney development. And after that, you worked as a postdoc at NIMR, National Institute for Medical Research in London, before actually starting your own lab at Oxford as a Wellcome Trust Fellow, which, of course, is an incredible program in itself. I mean, so your career trajectory has spanned a bunch of continents, a bunch of oceans. So before we let you go, we wanted to, ask you about your some advice that you might have for trainees hoping to follow that similar path of training at different diverse locations internationally. I think that's something that's perhaps becoming more and more commonplace in modern science. So could you talk a little bit about your road, your path, and what it's like training internationally like that?
Yes, absolutely. So in terms of advice, I mean, I've, I guess, always been just very, very curious about developmental biology, and I let that lead me. And it may sound odd, but I remember even probably when I was 11 or 12 years old, walking with my father from the eye doctor, the optician, and we were talking about what I might want to be. And it's weird. I think I remember then that I wanted to be a developmental. I wanted to. I didn't know that it was called developmental biology, but I wanted to understand how shapes came about, because particularly from the context of plants, they're really very funky. Leaves can be of different shapes. They can give you tendrils. They can give you roots can be in the air. The amount of morphogenetic diversity in plants is stunning, and I wanted to try to understand how that came to be. So I've always wanted to be a scientist to understand how morphogenesis. In terms of moving around, I think it's been, again, I've never been felt rooted in any one place, and so I was always happy to move around and experience different cultures. In terms of advice, I think maybe one bit of advice I would feel I might be able to give is actually to be very open. Sometimes, especially now in the competitive world that we live in, sometimes you feel that you can't share your exciting data with people pre-publication. But I feel that it's always helpful to be able to talk about them, because you never know where you're going to get really useful suggestions from. So that I've always found. I mean, and in the end, ultimately, it also keeps you going to be able to talk about. That's part of the reason I think we do this stuff, right? The sense of discovery, the sense of excitement we get, and of course, it's when you communicate that, it feeds back on itself and keeps you going, because it is obviously, we all know it's not always easy. Probably, I don't know about you guys, but the majority of my experiments used to fail, and you need to just keep on at it. And so, yeah, in terms of advice, I would say that try to be open about talking with people and being receptive to ideas, and you'll find that people, people are as trustworthy as the amount of trust you put in them. Is my view in a sense, and so that would be one thing. In terms of moving around, yeah, I've moved around a lot, and I don't think it's been bad for me at all. I found it very useful to be in different cultures and experience the different things. I loved my time in New York when I was a PhD student, but then I also loved really my time in as a postdoc in Mill Hill in New York, in London, in North London. And it was a time, it was in a sense the heyday of the Mill Hill is like, used to, particularly back then, it used to be like a mecca for developmental biology. It had the, big names, Rob Krumloff, Jim Smith, Rosa Beddington, and etc. And it was really a great place to be. And so, yeah, it was really enjoyable moving around. Now I haven't moved around for the past. I've been here since 2004, so it's been a good. It's the longest I've been in my life in any one place ever. Um, and then practical things come in. We have a family, etc. But if you're able to, certainly, I think it is helpful to be able to experience different things. Also, I did a PhD on kidney development and then moved to doing a postdoc on early embryogenesis. And I think it helps. Maybe some people would say that's not a big enough move. There was still the mouse. That was the common thing. I didn't change model organisms, but I think it's useful to be able to explore different systems, different questions. You get to learn different things and different perspectives.
That's three great pieces of advice that I really love, the staying open in particular, because yeah, I mean, no one ever publishes all those failed experiments, right? So sometimes the conversation is the only way you get at what people have tried before. So I really appreciate that. And, keep moving if you can. And finally, if you are going to put down roots, do it at a place that has a millennium, a pedigree, because you're likely to do good work there.
There are lots of good places. There are lots of good places.
Just kidding. You don't have to settle down in Oxford, people, but you could do a lot worse. Before we let you go, Shankar, we got a couple of science peripheral questions. We'll start with, you alluded to how you were born to do it. 10 years old, knew he was going to be a developmental biologist, even if he didn't know what the name of the job was, he knew what it was. But if you weren't a developmental biologist, not a scientist at all, what do you think you'd be doing with your life?
I've always wanted to be an astronaut. It's kind of corny, but I like sci-fi, I like space, and the wonder of being able to see the Earth from the outside, and from a slightly more geeky perspective, what that does to development of embryos, etc. Of course, there are experiments running on those type of things, but yeah, I would have liked to have been an astronaut, I think.
Well, here I am in the minority on this show as the only scientist that's not aspiring to the stars. As I'll tell you, he's
Why so, Aaron? Also,
yes, he does experiments in space. So yeah, don't.
Cool.
Yeah, I dabble.
Cool.
Finally, what is one hobby that you always wanted to pursue but were never able to?
I guess it's kind of linked, but astronomy, maybe not even just a hobby, as a career. Again, I have always been fascinated with space, the physics of space, etc., and or of how stars, the nuclear fusion, how they evolve, how they change, or how solar systems form, etc. So planetary physics, etc. But sorry, that's not a hobby. That's the vocation, I guess. But I don't think my physics, certainly when I was younger, and my maths were strong enough that I felt comfortable going into them. But certainly astronomy as a hobby where you can see the beauty of the stars. I wish I had more time. I made more time. Of course, it's not always clear here. It's a bit cloudy. It's often very cloudy, but I wish I could have made more time. I used to have a telescope as a kid. And I've seen the moons of Jupiter through it, and I've seen the rings of Saturn, etc. It was a very tiny telescope, but yeah, I think that would be one hobby I'd like to have spent more time on.
Well, Shankar, I gotta say, don't sweat the maths, partner. You still got a future in the stars. Take another sabbatical instead of India. Head over to California. Arun will set you up. He knows some people. Get your experiments up into space, partner. It's something you may enjoy for the next chapter.
I might do that.
Anyway, I really appreciate you sharing your, both worldly as well as extraterrestrial aspirations, and also your experience, the diversity of it, and all the insights. from your work. So, Shankar, this has been a really great conversation. On behalf of Arun and our listeners, we thank you.
Well, I just want to thank two of you for a really fun conversation. It was really great.
Our pleasure, always. All right, you guys, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on X at StemCellPodcast or by email at info@stemcellpodcast.com with feedback or to suggest guests. Till the next time, thank you guys so much for listening.